And she has been collaborating with some of our faculty here for quite some years, including Dr. Lin, Dr. Michael Bang. Today, her talk is use of superoxide dismutase mimetic during prostate cancer radiotherapy.
Thank you very much. So the focus of my lab is really trying to mitigate the normal tissue injury that occurs during radiation therapy for prostate cancer. So radiation is used, as we all know, very frequently for prostate cancer, and there's a very high survival rate. However, there are a lot of side effects that occur due to radiation exposure, and this is mainly due to the fact that the prostate is in close proximity to the bladder, the rectum, and the penile tissue. And this results in issues with bladder incontinence, rectal incontinence and diarrhea, and also erectile dysfunction. And currently in the clinic, there's really only one protocol that's used to help to prevent some of these side effects, and that is to use a hydrospacer gel, which basically removes the rectal wall from the prostate. And so this limits the dose that is actually given to the rectum. But this doesn't actually help to protect the bladder or the penile tissues or these nerves that innervate those organs. And it also only about 50% of prostate cancer patients are even eligible for this therapy. So there really needs to be a better way to mitigate some of this damage, because these patients are living a long time after therapy and they're suffering from these consequences. So free radicals are one of the major causes of normal tissue injury in prostate cancer patients receiving radiation therapy. So any cell, whether it's a normal cell or a tumor cell, is made up of water, and radiation is likely going to interact with that water molecule and produce water lysis. And this creates this hydroxyl radical, which is very damaging. In the normal tissues, we don't see as much direct damage or killing, but what we do see is a response to these cells because they are under stress, and they actually upregulate these enzymes called NADPH oxidases, and these cells produce free radicals in response to this stimuli. And so we see an increase in superoxide and or hydrogen peroxide produced in these normal cells. This actually stimulates TGF-beta, which causes fibrosis and tissue injury. Now, if you look at the tumor, what we see more direct DNA damage. This is caused by two factors. One is obviously the tumor has a higher dose achieved, and also those tumor cells are dividing more rapidly. So that DNA is more vulnerable, and we see more DNA damage, and this reduces the tumor size. And this, any surviving tumor cells grow back, and they are more aggressive, and they actually produce more superoxide, and this leads to more vessel regrowth and also tumor survival. So we postulate if we're able to remove superoxide from the normal tissues, we'll be able to inhibit this fibrosis, and in the tumor, we'd be able to inhibit this tumor regrowth. So we have a drug that mimics the active site of superoxide dismutase, which is the only enzyme in your body that can directly scavenge superoxide. And so just a picture is shown here. This particular drug has passed all the safety toxicology studies, and a new investigational drug has been filed with the FDA, and we have a completed scale-up synthesis of GLP quality drug at hand. Currently, this drug is in phase 2 clinical trials as used for array protector in glioblastoma patients. And then we have a phase 2 trial for head and neck cancer we actually are contributing at UNMC. And then we have a phase 1 clinical trial in anal cancer also at UNMC. And so we're wanting to see if we can also use this compound for prostate cancer. So a little bit more on the chemistry of this. So BMX, as I mentioned before, scavenges superoxide. And in normal cells, we don't have as much superoxide being produced at basal levels. When BMX is added, it will convert that superoxide into hydrogen peroxide. And these normal cells have a very robust system to get rid of hydrogen peroxide. These are called hydroperoxidases. And this will quickly convert that hydrogen peroxide into water. Also, superoxide will interact with nitric oxide and produce another very damaging radical called peroxynitrite. But because superoxide levels are lower with BMX around, we have more NO and less peroxynitrite. So this is not a damaging radical. In cancer cells, what we see is at baseline, we have higher levels of superoxide. When the drug is put in here, we see a large conversion into hydrogen peroxide. But cancer cells have a very poor system for removing hydrogen peroxide. Their hydroperoxidases are not very active, and therefore we see an accumulation of this hydrogen peroxide, and it's not converted into water. Additionally, we see that there's, again, more superoxide around, and so this will actually interact with that NO and produce this damaging peroxynitrite radical, which causes damage in the cancer cell as well. So what we actually observe is we see an overall decrease in ROS in normal cells when treatment with this drug. But in cancer cells, we see the reverse. We see more ROS being produced, mainly this hydrogen peroxide and peroxynitrite. And to further confirm this, this is some data that we collected in our lab. This is looking at cancer cells and normal prostate fibroblasts. And the last two columns are treatment with the drug. And you can appreciate that we see a lot more hydrogen peroxide being produced in the cancer as compared to the normal cells. So this led us to a new hypothesis that adding in this SOD mimic will scavenge that superoxide in the normal tissues and inhibit this fibrosis pathway. But in the tumors, we will scavenge some of that hydrogen superoxide, but it's going to be converted into hydrogen peroxide, and this accumulation is going to result in tumor killing. So I'm going to show you some data that we've collected over the years, looking at some of these target organs in pelvic irradiation and showing that the drug is able to protect from both acute and chronic irradiation damage. So this is work that was done in combination with Dr. Chi Lin here at UNMC, where we use a Linac irradiator and we radiated the rectum with 20 grays of irradiation, and then we harvested these tissues at 9 days post-irradiation. And what we noticed is every animal that we irradiated by 9 days had diarrhea, as shown here, and also had hair loss around the anal region. And treatment with the BMX compound, we didn't have any diarrhea in these animals, and we also see that this hair is intact. When we pulled the rectal tissue out, and we actually formed histology on there, so you can appreciate that the radiated bowel had epithelium that's sloughed off. We have apoptotic crypts indicating that this tissue is remodeling after damage, and treatment with the BMX compound, we see this was largely protected, and the damage scores is shown over here on the right. We also wanted to look at some chronic side effects, which of course is fibrosis. And so we performed another experiment where we irradiated the pelvic region of these mice. We gave 7.5 grays 5 days in a row, and then we let the animal sit for 6 months, and we treated with and without the SOD mimic. And we stained the bladder tissues with trichome stain, and this will stain any collagen blue. And so the red is the smooth muscle layer in the bladder. And what you can see is in both the control and the drug treated, we don't see very much of this blue fibers in the muscle layer. But in our irradiated cohort, you can see that there is a lot more of these fibers present in this smooth muscle tissue, and treatment with the SOD mimic protected this, and this is quantified over here with I think an N of 10 for each group. We performed the same experiment, but we looked at erectile functionality in these animals before we sacrificed them, so that we measured intercavernous pressure as a direct indicator of erectile functionality. And you can appreciate that radiation causes significant reduction in erectile function, whereas treatment with SOD mimic helped to protect from this. So we have data that we're able to protect the rectal tissue, the bladder tissue, and the erectile tissue from damage due to radiation exposure, both chronically and acutely. So how does radiation manifest after, or how does fibrosis manifest after radiation? Well, we think that reactive oxygen species play a very large role in this. So with radiation, these cells become more oxidatively stressed, and they become epigenetically modified. And so these normal fibroblasts will turn into an activated fibroblast. And these activated fibroblasts are the ones that secrete this extracellular matrix, which causes this fibrosis or scar to form. And TGF-beta is the signaling molecule that really helps to drive this process. So we were interested in just demonstrating that that these drugs actually do scavenge superoxide. And so we measured superoxide in normal fibroblasts treated with and without irradiation, and we measured this 24 hours after these cells were exposed to irradiation. And you can appreciate that with radiation, we see about a 1.5 to 2-fold increase in superoxide production, and treatment with the SOD mimic blunts this increase. So we are scavenging superoxide in these normal cells. TGF-beta signaling, again, is another pathway that's activated in these fibroblasts. And so this is a little schematic. Basically what happens is you have a TGF-beta ligand that is bound to this latent protein. When radiation hits the cell, this becomes decoupled, and this TGF-beta then can bind to the TGF-beta receptor. And then this can phosphorylate these proteins called SMAD proteins. So we have SMAD2, SMAD3 complex over SMAD3, SMAD4 complex. And these proteins can then enter into the nucleus and act as transcription factors to upregulate profibrotic and senescent genes that are responsible for creating fibrosis. So we looked at some of these different components of this pathway with and without drug treatment. And what you can appreciate here is looking at TGF-beta receptor 2. When we treat with the SOD mimic, we see a significant reduction in the expression of that receptor. We also see a significant reduction in the SMAD proteins. So we see for SMAD3, this is significantly reduced. The phospho-SMAD2 and total SMAD2 is also reduced with the manganese porphyrin treatment, and SMAD4 is also significantly reduced. Interesting, SMAD7, which is a inhibitory molecule of this TGF-beta signaling pathway, was largely unchanged. And then a downstream target of this SMAD signaling pathway called plasminogen activator inhibitor 1, or PI1, was significantly downregulated with treatment of the SOD mimic. So this data highly suggests that treatment with the SOD mimic is blocking this TGF-beta signaling pathway. So I hope I've convinced you with these data that I just showed you that in the normal cell, treatment with the SOD mimic is able to block this TGF-beta signaling, block superoxide, and then prevent fibrosis. But what's going on in the normal cells? The cancer cells. And so again, we predict that that we will see this increase in hydrogen peroxide, and this will help to lead to more tumor killing, especially in the presence of radiation. So we performed an orthotopic prostate cancer model where we injected either LNCAP or PC3 cells, and of course, LNCAP is our androgen sensitive, and PC3 is our androgen insensitive cell line. And we treated these animals with radiation, with 2 grays of radiation 5 days in a row. And then we harvested these tumors 2 weeks after radiation. And you can see that drug treatment alone did not really cause much of effect here, but in the presence of irradiation, we see a significant reduction in these tumor sizes. We also measured the cell proliferation of these tumor cells. And again, treatment with radiation and the drug helped to significantly reduce those levels. And if we look at overall survivorship of these mice, the mice that received both radiation and the drug in both cohorts were the ones that lived the longest. In addition, we also measured metastasis of these prostate cancer cells. So this is a syngeneic mouse prostate cancer cell line. And when we treat with radiation or radiation plus the SOD mimic, you can appreciate that we see less metastasis as well. So from these experiments, we can show that the drug is not protecting the cancer cells from irradiation, which is obviously very important before we put this into patients. To further support our hypothesis that this drug is acting as an antioxidant in normal tissues and a pro-oxidant in cancer cells, we took the orthotopic cancer and we collected the prostate and the tumor connected to that. And we then stained for lipid oxidation, which is a marker of oxidative stress. So the panel on the left box here is cancer cells, and the panel on the right is the normal prostate tissue that's adjacent to the tumor. And we immunostained for 4-H&E, which is 4-hydroxyneonol, which is a marker of lipid peroxidation. And what we see is, and with both PC3 and LNCAPs, when we treat with radiation and the drug, we see a significant increase in this lipid oxidation in our prostate tumors, which again is indicative that we're creating more oxidative stress in these tumor cells, and this could is likely leading to the killing of these tumor cells. But in the normal tissues, what we see is radiation causes a nice robust increase in lipid oxidation, but treatment with the SOD mimic helps to blunt that. And so again, this data shows in the same animal, in the same model, that we're acting as a pro-oxidant with the tumor and an antioxidant in the normal cells. Finally, a lot of prostate cancer patients are treated with androgen deprivation therapy along with radiation. And so we wanted to determine what is the effect of this drug in combination with androgen deprivation therapy. So this is in vitro clinogenic assays that were performed on either PC3 or LNCAP cells. So this is just with radiation and drug. You can appreciate the drug by itself causes a reduction in tumor growth, but in combination with radiation, we see a significant reduction. When we throw ADT into the mix, what you see is the drug is now almost as potent as radiation alone, which was kind of unexpected. And then treatment with the drug and a radiation in ADT, we see a further significant reduction in growth. And to further confirm that this is actually due to dihydrotestosterone and not something else, we added DHT back in, and you can see that this rescued the cells from what appeared in B back to more of what we saw with A, and that we lost the efficacy of BMX as a potent radiosensitizer. And then in LNCAP cells, we see a similar finding. So again, without any ADT, the drug by itself causes a significant reduction. In combination with radiation, we see a very substantial decrease in clinogenic survival of this tumor cell. And then when we throw ADT in there, this is even more exaggerated. So we're really excited about this piece of data because this, I think, shows that potentially in our patients that are receiving ADT, we might even see better radiosensitization in this cohort of patients. So finally, we would like to start clinical trials here at UNMC for looking at the effect of BMX in patients that can't undergo hydrogel spacer replacement as a radioprotector. And so this would be patients with extracapsular extension or seminal vesicle invasion prior to prostatectomy, or potentially patients with high-risk disease, or patients undergo salvage radiation therapy after radical prostatectomy. And to note that these patients are the patients that have the highest risk of reoccurrence of prostate cancer, and these are the patients that will actually die from prostate cancer. And so we think that this cohort would be very good to actually look to see if we put this drug in, do we actually help to promote progression-free survival in these patients. So we've written several grants the last couple of months here. So hopefully one of these will get funded. We're proposing to use a randomized placebo-controlled phase 2 clinical trial to determine if BMX is an efficacious radioprotector and hope to enhance overall quality, patient quality of life. So our primary endpoint would be acute GI and GU toxicity, and then our secondary endpoint to be progression-free survival and the chronic complications as well. And with that, I would like to acknowledge all the people that helped me do this work. This is my laboratory shown here. I'd like to thank Dr. Kosmachek, Dr. Chatterjee, and Dr. Srishamel for all of their work, as well as Yushen Zhu, who's a graduate student in my lab, produced most of the data that I showed you today. And I'd also like to acknowledge my collaborators, in particular, Dr. Bain, Dr. Ink, Dr. Lin, and Dr. Sikong Lee. And everybody else listed here as well for all of their help in getting this project up and going. So thank you.
All right, thank you, Dr. Deegan. All right, our next speaker is Dr. Tatiana Brownish. I consider her to be my mentor for the past 4 years. She's currently the professor of Department of Pharmaceutical Science at our institutes. She's also the co-director of drug development and nanomedicine here. Her talk today is nanotechnology for radiation oncology.
Thank you. So good afternoon, and of course I would like to thank the organizers for their kind invitation. But most of all, I would like to thank organizers and all speakers for absolutely great opportunity which the symposium present for myself, for my education in this area. And I would like to share